Welcome back. And so there's always that one person on the team who seems to be able to play every position, and that's our next speaker, Dr. Benjamin Levy. He seems equally comfortable if he's talking about targeted therapy or immunotherapy or clinical trials. He just is one of those people. And so he's actually the MVP of this year's International Lung Cancer Survivorship Conference because he has not one but two presentations. He has a on-demand session in our Lung Cancer Basics, which really focuses a lot on targeted therapy or precision medicine for lung cancer. But as promised, now up next, he's going to be talking to us about antibody-drug conjugates. And so it's a pleasure to welcome ILCSC's MVP. He's going to tell us about the ABCs of ADCs. So welcome, Dr. Levy.
Thanks, Amy, for that generous introduction. I like utility player better than MVP. It shows me my versatility of moving from different positions. But probably my favorite position in the game is with antibody-drug conjugates. This is clearly a new class of therapies that we're really just beginning to scratch the surface on. These drugs, as I'll go through, have landed front and center in breast cancer. And for the first time in a while, we're borrowing some of the therapeutic strategies from breast cancer and applying them to lung cancer. And the title of my talk are these diamonds in the rough? And there's I think a lot of controversy about these drugs. I think that clearly we're making strides with some of these, but others we're asking a lot of questions. And I'll kind of go through what I think those questions are, and I'll go through where I think we're heading with these compounds. And so it used to be that when I talked about lung cancer to my patients or my fellows, I would say, look, there's really three types of strategies to treat lung cancer. There's targeted therapies, and we all know about those. There's immunotherapies, which are very different than targeted therapies. And then there's chemotherapy. And now my narrative is a little bit longer because now there's antibody-drug conjugates. So that's a fourth category of strategies. And I think this has been really a win for patients because it continues to move the field forward with these novel compounds. So what is an antibody-drug conjugate? For those of you who are uninitiated with these drugs, that's okay. Please don't feel like this is going to be a super science talk, but this is a pictorial that does a really nice job of capturing what these compounds or how these compounds are constructed, and also what are the elements of these compounds. So the compounds really they have three components. They have a monoclonal antibody, which is this blue-green molecule you'll see here. And we all are pretty familiar with monoclonal antibodies, all the way back to a drug called Herceptin in breast cancer or Avastin in lung cancer. And this monoclonal antibody then has a linker. That's the gray little area down below there. And that linker then attaches what we call a warhead or a payload or a chemotherapeutic agent to the monoclonal antibody. And so essentially the whole goal here is if you have this monoclonal antibody and the green part of that monoclonal antibody is able to seek out a protein that's only on the cancer cell and not on the rest of our cells, this is truly targeted chemotherapy, where sort of a javelin missile, where this molecule can essentially be infused. It's not oral, it's infused. The molecule is able to get through the bloodstream, and then that green area of the molecule begins to seek out those proteins only relevant on the cancer, attach to those proteins, and then unload the warhead or the chemotherapy. And so really we're trying to selectively deliver chemotherapy just to the cancer and not to the rest of the body. And it's a really fascinating concept that we're doing here. This seems pretty straightforward. It's not. There's a lot of nuances and a lot of details here that I can't go over that become incredibly complex and this is really about sophisticated biochemistry and how these and biosynthesis of how these drugs are really made in the lab, and what type of chemotherapy you're going to add to the monoclonal antibody. Nevertheless, a fascinating story, and one that continues to evolve in our field. Again, these drugs really have been more common in breast cancer, and more recently, we're now learning about them in lung cancer. And we're a little bit behind the other tumor types, but that doesn't mean we won't excel. We will. We always do compared to other tumor types. So I thought I would go over this a little bit here. So we talked about this monoclonal antibody with that green part there. And you really want that green part of the monoclonal antibody. Let's just go back to it one more time. This green part to seek out the protein, the antigen, okay? And what you obviously want is you want a protein that is selectively expressed on the cancer cell, but not anywhere else, right? If it was expressed everywhere, there would be exceptional toxicity because the monoclonal antibody would just go to all cells. So you want that antigen to be only on tumors, not normal tissue, and you want a lot of it. You want a lot of protein on that cell, because the more protein you have on that cell, the more likely the antibody can find it. And so you want that protein accessible to the antibody. And then the third thing that's just so important, and again, this is where we as clinical researchers and translational biologists and clinicians are trying to understand, is that it's not enough for the monoclonal antibody just to attach to the antigen. There has to be something called internalization, which is essentially that antigen captures that protein, captures that monoclonal antibody with the payload, and then moves it inside the cell. And that by itself is an extraordinarily complex event, and one that we're still trying to understand as to why some antigens or proteins that are only on the cancer cell, why is it that even when the antibody or the javelin missile attaches to that protein, why does it not get internalized? Because you need that whole construct to internalize into the cell so that the payload or the warhead can be released. And so we've got a lot of work to do with these compounds, but these are the things that we want out of an antigen. There are multiple targets in lung cancer for antibody-drug conjugates. And again, that bottom kind of glow of hue of orange or red or whatever it is inside the cell, and then everything that you're looking at is outside the cell. And again, these proteins like HER2 on the left, HER3, then TROP2 and CCAM and CMET, B7H3, these are all those proteins, those antigens, those selective targets that only exist, hopefully, on the cancer cell and not on the rest of the body, the rest of the host tissue. And the whole idea, again, is, as you can see, these particular agents, and they have funky names like adatrastuzumab emtansine and patritumab deruxtecan, which has taken me a while to learn how to say, is those compounds are really trying, again, to seek out these antigens. But, as I mentioned to you, just seeking out that antigen or that protein that sits on the cell surface is not enough. You've got to have then this marriage of the antigen and the antibody, and then complete internalization of this whole complex, because the chemotherapy payload can't get into the cell or can't release itself until it's inside the cell. So a lot of nuance here, and this is something that we're trying to desperately work out, how these drugs work and what's the best way to use them and who to use them for. So how do these drugs truly work? This is, again, we really wrestle with this frequently, is, do we consider these drugs chemotherapy? Because, they have a chemotherapy payload, that little star, yellow star that was attached to the monoclonal antibody, that's just an old school chemotherapeutic drug that's attached to the monoclonal antibody. Or do we view this as targeted therapy? I mean, I just showed you this picture of, like, these monoclonal antibodies coming in and attacking to a specific target. That essentially is that is essentially targeted therapy. So which is it? Is it chemotherapy or is it targeted therapy? And my answer is always to this question, yes. So they're probably both. Is this chemotherapy version 2.0? Is this targeted chemotherapy? I think we're still trying to understand again the nomenclature of these compounds. So I think it's really complex. And then again, this is the best pictorial I can give you of how these drugs we think work. You see up there the ADC, the monoclonal antibody, and in this sense, the construct, the red is the payload or the chemotherapeutic agent. The monoclonal antibody, the light blue part of that monoclonal antibody, selects or picks out and attaches to an antigen that is specifically located on the cell surface of a cancer cell, hopefully not a normal host tissue cell. Once that binding occurs, again, there is this internalization, and you can see that here. So number one is ADC binds to the antigen receptor. Number two, internalization, the most important point. Once internalization happens, then and only then, generally, can the payload be released. And once the payload is released, it causes cell kill, right? It's going to damage the DNA, and you're going to get what's called apoptosis or death of that cell. The nice thing about these drugs also is when the payload is released, not only does it kill that cell, but it also kills neighboring cancer cells. So you get what's called this bystander effect, where these payloads diffuse through the whole tumor, even if some of the tumor doesn't have that particular protein in which the monoclonal antibody is trying to seek out, that payload will leak and kind of capture all the cancer that's nearby. And that bystander effect is really, really important for us, for cell kill. So, in terms of the ADCs that we look at, and I know Dr. Liu kind of gave you a primer on ADCs as well, but the first place that ADCs have started in lung cancer has been on the heels of the molecular revolution of targeted therapy. So I think for now, you probably are aware that lung cancer can be divided up into these distinct molecular niches. And, incredibly and thankfully, we've got different drugs for different genes, right? EGFR genes, EGFR exon 20 genes that have drugs, MET exon 14 skipping genes that have drugs, ROS1, ALK, all of these have drugs. And one of the other ones that now we've been able to isolate in lung cancer that's also seen in breast cancer is HER2. HER2 is yet another mutation that we see in lung cancer roughly 2 to 4% of the time. And it's mutually exclusive from all the other genes. So if you have HER2, you don't have any other of these slices of the pie. And that's really where ADCs begin in lung cancer, is in the HER2 mutated space. Now, historically, before this drug, we tried so hard to target HER2 with all these pills that were, also used in other types of cancer, and it just didn't seem to work all that well. Now, we now know it from World Lung just 2 weeks ago or a week ago in San Diego, there's some fascinating HER2 molecules coming out that are pills. But for now, we have a great antibody-drug conjugate that's approved. Once again, a monoclonal antibody with a linker, with a payload, a very high payload. And I don't want to go into the nuances of what DAR means, but DAR is essentially this abbreviation, drug to antibody ratio, which talks about how loaded that monoclonal antibody is with chemotherapy. And 8 moieties per one molecule is pretty darn loaded. So it's, very potent. And I'm not going to go through all the data with this, but, for those of you who have seen waterfall plots before, you can see this and say, wow, that's really good. I mean, this is an incredible response from a drug. For those of you who haven't seen a waterfall plot before, each bar is a patient's size of their summation of the size of their tumors. And downward bars mean that's how much the tumor shrank, the sum of the tumor shrank when given this drug. And you can see for most of the patients, everyone's tumor shrank to some degree when getting this drug. In fact, there were only 2 patients in which the tumor grew. So this is what I call an encouraging waterfall plot. and one where this led to the approval of trastuzumab deruxtecan, the antibody-drug conjugate that I just showed you, specifically for patients that are HER2 mutated. And that again underscores the importance of testing. It's so critical that we test, don't guess. We need to use liquid biopsies and tissue biopsies to make sure that we have this. It all started, of course, with EGFR and ALK and ROS and RED and KRAS now and exon 14 and others, Interact. But HER2 now is front and center as well. And we need that information when we give this drug. We can't give it unless they're HER2 mutated, or very confusingly, and I don't want to get too much into the nuances here, trastuzumab deruxtecan, the ADC I just showed you, is also approved for patients who have lung cancer who have not HER2 mutations, but HER2 overexpression. The protein is overexpressed on the cell surface, which is different than the mutation. That only occurs in around 5 to 10% of patients, but nevertheless, that also should be done. We need to test for HER2 as a mutation, but we also need to test for HER2 overexpression, which is a different thing. So this is trastuzumab deruxtecan at work. This is one of my favorite patients who came in with a cough and shortness of breath. We did a liquid biopsy on this patient because he was having so many symptoms, and of course, liquid comes back in such a quick time. So we got the liquid biopsy back in 5 days. He was HER2 exon 20 mutation positive. That's the most common type of HER2. 2 doses of trastuzumab deruxtecan, 2 doses, and voila. You see the tumor has essentially melted away. And this is what we expect from targeted therapy. This is what we expect from really good high-performance drugs that are selective for the tumor and really don't touch the rest of the body. So it's an incredible story. And look, this is not a one-off. This is something that we see all the time. And the patient feels great. And you know, it's great to look at these images, but it's also great to see the patient every 3 weeks and talk to them about other things, not their cancer, which is what these drugs allow us to do in our clinic sometimes, which I love. So I just, again, this is what we see with some of these antibody-drug conjugates, and it's a really great story, especially for patients like this. So, you know, I don't want to get too much into the weeds here, but there's a lot of questions I get about, okay, these are new class of compounds. You know, we've all heard the story about how targeted therapies, these small molecule inhibitors, these pills like Tagrisso, like crizotinib, like alectinib, like lorlatinib, all these drugs, they do a great job not only tackling the cancer below the neck, but they also do a great job of penetrating the brain because they're so small. They can actually get inside the brain and shrink the tumors there if there are metastases in the brain. And when these drugs came out, we never thought that these types of drugs would actually get into the brain. They're just too big. But what was really interesting about some pooled analysis from some of these studies is that, you know, there were response rates as high as 50% of patients had intracranial responses, meaning their lung cancer that had gone to the brain, they were treated with an ADC like trastuzumab deruxtecan, and lo and behold, the tumor shrank in the brain as well. And you can kind of see this again on these waterfall plots on the right here. These are patients this is how much tumors in the brain from the lung cancer shrank with, yes, some of them had gotten radiation, but some of them hadn't, who were given this drug. So, you know, we're learning a lot that these drugs not only do a great job in the chest and the lungs, which I showed you, but also have a wonderful potential ability to get into the brain. And this is where things get so nuanced, because if you would have asked me, you know, when we started looking at these drugs in clinical trial, hey, what do you think that's going to happen in the brain? I would have said, there's no way these drugs are getting into the brain. They're just too big. But here we go. We see clinical observations that run counter to what we believe, which is a good thing. I want to mention 2 other. So there's a lot of ADCs that are coming out. Trastuzumab deruxtecan clearly is the poster child for ADCs and lung cancer, as I just showed you. There are 2 other drugs that you're going to hear a lot about come January, February 2025. And these drugs, sacituzumab govitecan and datopotamab deruxtecan, are 2 ADCs that have been tested in lung cancer that have now shown us results really only in presentation form. We don't have the final manuscript yet, showing that these drugs are good. They may not be great, but they're good in the second line for patients without an EGFR or any sort of mutation, patients who've received chemotherapy with immunotherapy with advanced stage disease who have cancer growth. That's where these drugs were tested. And I think we would have liked to have seen better results from this, but nevertheless, it seemed like there was a benefit with these drugs compared to the standard of care. Whether the FDA will approve these drugs or not, it's unclear. These drugs target a different antigen called TROP2. Before the ADCs came out, there wasn't a lot of understanding about TROP2 expression. This is, again, an antigen that sits on top of the cell surface. And I think, to be transparent, honest, these drugs may have underperformed, but nevertheless, they may represent another therapeutic option that we can kind of, take from our armamentarium and give to some patients. And so I just ask you to be on the lookout for these two compounds because the FDA is going to be reviewing them. The other drug that I didn't want to get too in the weeds, so there's these two, there's trastuzumab deruxtecan, there's these two agents that are TROP2, and there's one more agent that's gotten a lot of traction, which is patritumab deruxtecan, which is yet another antibody-drug conjugate that's being leveraged in the EGFR space. And there's been a lot of conflicting data about this drug. This drug looked like it was going to be approved as a third-line agent for EGFR-positive patients based on some preliminary data. And then, unfortunately, there were issues with the factory manufacturing of the compound, which, again, tells you how critical, the biochemistry of these drugs is, the synthetic biochemistry and the formulations that go into making this drug. This drug was about to be approved, and the FDA said, not these two, but patritumab deruxtecan. And the FDA said there's manufacturing issues. So we'll have to wait and see what happens to patritumab deruxtecan, the other agent that I'm not showing you. But I think that this is just the beginning of these drugs. You're going to hear a lot more about these as we continue to delve into the science. I think it's important. These drugs are great, but we need to recognize the scope of common adverse events. And I always tell patients, look, there's two categories of toxicities with these drugs. There's your traditional chemotherapy side effects, which, again, remember, they have a chemotherapy payload on the monoclonal antibody. So undoubtedly, you're going to get some side effects that are chemotherapy related. So there is a level of alopecia, although not as intense as the regular chemotherapy drugs. There can be nausea, just like there is with chemotherapy. There can be liver issues, just like there is with chemotherapy. There can be fatigue, and there can be hematological issues. I would say that depending on the drug you're talking about, these tend to be less than some chemotherapies that we use in lung cancer. I would say Alimta is still a really well-tolerated drug. And so, how to leverage some of these side effects or look at some of these side effects through the context or through the prism of what Alimta is able to do in terms of managing, in terms of not being so toxic. This is where I think we're still trying to learn. And then on the right here are sort of these, what I call the adverse events of special interest. And these are really just relevant to ADCs. And we're learning about them. Number one is ocular events. We're starting to see very strange things, dry eyes. We're starting keratitis. We're starting to refer these patients to our ophthalmologist to help us manage these. It tends to be not so severe, but something to look out for. Stomatitis, which is this inflammation of the mouth, is something that we see with one of the TROP2 antibody-drug conjugates that I just showed you, datopotamab deruxtecan. And we have to learn how to manage this and mitigate it and be proactive about it. Cardiac events are very rare, but with trastuzumab deruxtecan, the drug I just showed you, that's in the HER2 space, there is a small chance of risk to damage to the heart, although it tends to be less than 1%. And then pulmonary, these drugs, just like immunotherapy, can cause interstitial lung disease. And so we have to be mindful of screening patients, looking for signs, making sure we assess symptoms so that we can treat patients appropriately. cough, worsening cough, shortness of breath, fevers, all these things that can happen, even in the context of lung cancer, having lung cancer, can also occur as a consequence of the drug. And so we have to be very mindful. And I think, again, I always talk to fellows and residents about this, that look, two separate categories here, but remember these adverse events of special interest, we really need to keep our eye out for them. What's next for these compounds? And don't let this intimidate you, but this version of antibody-drug conjugates that we currently have is just the beginning. It is just the start. And I'm going to be as transparent and as critical as I can, and say that in 5 years, the ADCs that we currently have will no longer be around. There's just no way. The science is too fascinating. There are pharma companies now doubling down and creating industries or factories all over the world dedicated to constructing these types of agents. And they're going to be very different moving forward. And I'll just mention a few of these because there's going to be so many different iterations and permutations of how these drugs are constructed. And remember, it really is sophisticated biochemistry that put all these things together. One of the ways that is going to change things is that instead of having one chemotherapy type only on the molecule, there'll be two separate chemotherapies. So you'll be able to knock out the tumor in two separate ways, all by delivering just one monoclonal antibody. That monoclonal antibody will have two separate chemotherapeutic agents attached to it. So it's sort of a double hit, 1-2 punch. And I think it's going to be really interesting. The other thing that's going to happen is they're going to have these constructs, these monoclonal antibodies that don't have a payload on them. But essentially, they'll bind to the antigen on the surface of the tumor cell. And then once it binds, they will completely engage the immune cells around that tumor to swallow the tumor. And that's kind of what immunotherapy does at a level. And this will also be kind of next level. And then the one that I love the most is called pro-body conjugates, which is essentially that, we still deal with toxicity of these drugs, right? These drugs, in a perfect world, would just go to the tumor and never go to the host tissue. But it's not perfect. It never is. And so one of the things that is being done is basically creating what's called a masking peptide, which is essentially, and I'll just kind of throw the visual up here, is that when the monoclonal antibody comes into the contact of a regular normal cell in the body, not cancer, that masking peptide will not allow it to engage in normal tissue. Only when it is engaged in the cancer cell will the masking peptide release itself and allow the monoclonal antibody then to deliver the cytotoxic agent. It's really neat and really selective and really nuanced and really precise. And so I'm really excited about how these constructs are going to be done. It is going to be very complicated, but also at the same time, very exciting. I mean, these are all the new, like if we look at even when I started in the field, 2011, 2012, how number of new ADCs, there were like 2 or 3. There were 0 in 2012. In 2016, when I moved to Hopkins, there were 4. Now look how many ADCs there are in 2022. There's more than 50. I would say in 2024, this is 2022, I would say in 2024, there's probably now 75 to 100. And so I do think at some point we are going to replace chemotherapy with these types of drugs because they're just going to be able to deliver higher, more potent agents with more ability to deliver payload directly into the tumor cell and have no offloading into the normal tissue. So very little toxicity. So, I'll end with my 2 favorite slides. I do this no matter what talk I do, as I am the utility player, not the MVP. But every talk ends with this when I talk to patients. And that is a testament to patient advocacy, a testament to the nursing, a testament to the physicians, a testament to the scientists, a testament to the pathologists that have really, and the social workers too, that have begun to make this cancer journey much more nuanced and much more personalized. So this was the cancer journey when I started in the field. There was prevention, screening, diagnosis, treatment, and recovery. And maybe there was some psychosocial, implementation along the way. And we now know that this is so much more complicated, and again, in the best way possible. We need to factor in, how the patient was pre-diagnosis. We need to factor in what are some of the alternative medications that we could integrate. We need to factor in the psychosocial issues here, the psychological element of this. We need to factor in financial implications. We need to factor in travel time to the infusion center. We need to factor in family dynamics. We need to factor in, novel clinical trials. It is so nuanced and personalized that this is my favorite slide. And again, I think we've gotten to this slide on the coattails of patients, because patients have said, "Look, I need this." They've been advocates and voices, and that has made this construct happen, I think. And don't get me wrong, there's been plenty of people in the field of medicine that have helped move this along. But I will tell you that I have taken a lot of my cues from patients early on in my career to say, okay, this works. Patients coming to me and saying, I went to acupuncture and it's the only thing that helped my nausea. And, after 4 or 5 patients come to me, I say, I need to partner with an acupuncturist in our system so that we can make sure these patients have access to it. And we've done that. So it is very, very important, the journey at hand. And then again, this whole idea of patient-centered care and shared decision-making that really puts together empathy and compassion is really important. It's hard for physicians to do this with everyone, but it really is important. This is not a straight line, this journey. It is, as such as life is, a roller coaster with turns and curves and dives and hills. And, we have to recognize those nuances of the journey. We have to factor in patient wishes. We have to factor in patients' family wishes. We have to factor in goals, needs, beliefs, all those things. And we have to keep our eye on the science at the same time. And how to do that, I think, is a skill set that I'm still learning how to do. Conclusion here, antibody-drug conjugates represent a novel therapeutic for patients with non-small cell lung cancer. Again, it's the antibody, it's the linker, it's the warhead. We are just beginning to scratch the surface on this. We're not really sure how these drugs work. I gave you sort of the schema, but I didn't want to get too much into the weeds here because we're still wrestling with some of the data that we're seeing. We need to define those patients that are going to most likely benefit from these drugs. We have that in the HER2 space. We know that it's patients with HER2 mutations that really derive a benefit from that ADC trastuzumab deruxtecan, otherwise known as Enhertu. But there are others that we're still learning. The future is bright. I mean, the sophisticated biochemistry that's going into this and trying to learn how to make these constructs tailored to the patient. It's more sophisticated, dare I say, than targeted therapy. Targeted therapy is sophisticated, don't get me wrong. You've got to develop a small molecule inhibitor that will go inside the cell and bind to the intracellular domain of that protein. This is a little different. It's a little more complex. We're going to start using these in the advanced stage setting first, of course, and then we're going to move them to earlier stages. So I think with that, I'll end, and thank you for allowing me onto your Zoom screen, and I'm happy to take any questions that are through the chat. And there's no such thing as a stupid question.
Thank you, Dr. Levy. That was an incredible overview of an increasingly important topic, the antibody-drug conjugates in lung cancer. And, when you started rattling off the drug names, I felt like you were doing some Harlem Globetrotter moves there, kind of showing off a little. But very good job with rattling off all those complicated names. So, I want to start with the most obvious question, because we've already had some conversation around, starting with Dr. Peters' talk this morning, the accessibility of targeted therapy in precision medicine. And, this is something that's not available to everyone around the world. When we look at our ability to do biomarker testing, even here in the US, we know that not everyone's getting access to that. And these therapies are dependent on our ability to detect the biomarkers so we know who's a candidate for these treatments. So, how do we think about getting these to the people? I mean, science is exciting, it's super cool, but if we can't get them to the people who can benefit, then what good is it? So how do we think about broadening the availability? You talked about global manufacturing of them, but again, they have to be delivered in an infusion setting, and
Yeah.
so it's a little bit more complicated than just delivering a pill. So I'm curious your thoughts. How can we
Yeah,
broaden
I think
this?
it is twofold. And one is, again, I've said this many, many times, is that we still are not performing to our highest level when we talk about testing for the genetic markers in the lung cancer. We do it okay, but we need to do better. We need to make sure that no stone is unturned so that we can identify the right gene or the right protein for targeted therapy and now for antibody drug conjugates, right? And so the whole idea here is that these are going to be infusions most likely, and it's going to have to be similar to the immunotherapy story, which is that at institutions at a very high level, no matter where you go, whether it's in Sheboygan, Wisconsin, or Austin, Texas, or wherever it is, that institutions at a very high level infusions are going to have to make sure they have access to these drugs if they're FDA approved. And this was the problem when immunotherapy came out at first. We're having a hard time getting them in our infusion space, and then we're having a hard time learning about how to give them. And so I say, one, it's testing to make sure we identify the right patient. Two is working at a very high level across the country to make sure that these institutions have these drugs. And then three, most importantly, is the education piece, which is, making sure that both physicians and patients understand the side effects, understand how to manage them. And this is where I hope people like me can come in, because when immunotherapy came out, it was the same thing. People were like, what's happening? There's inflammation in the lungs. And we didn't know at that point, oh gosh, there's an element here that can cause pneumonitis or inflammation of the lungs. So it's going to be all three of those, I think. It's a big part of this is going to be education, though.
Absolutely. And so we were just together at the World Conference on Lung Cancer in San Diego, and there were two presentations. You talked about the ADC and the HER2 setting, trastuzumab deruxtecan, and that it can unleash this complication of interstitial lung disease, pneumonitis. So that's something we have to be on guard for. But we heard two presentations which show great promise as kind of targeted therapy options. So I'm curious, how quickly are we going to see trastuzumab deruxtecan kind of continue to stand in the HER2 space, or is it going to quickly be supplanted by these different options that don't maybe require infusion? And what's that competition look like?
This is the question we've been wrestling with for a solid 14 days since we saw this data. I mean, HER2 mutations, have been front and center for antibody-drug conjugates, and again, they work really well, as I showed you. As I said in my talk, the reason why we were so excited about trastuzumab deruxtecan, this ADC, is because most of the targeted therapies we have tried for HER2 mutation lung cancer has been a big disappointment. And along come these two drugs that were just presented last week. One drug called zongertinib. I don't know how you come up with these names. And the other is a Bayer drug that doesn't have a name yet. And what we saw from these compounds was exceptional activity, these pills with exceptional activity with these agents. And so the question will be, will the antibody drug conjugates sort of go away in this space, or will it be a part of a treatment continuum where patients get in HER2 first, and if there's growth of the cancer, then they get the pill, or will it be reversed? I mean, one of the things that's happened here is that there's so many drugs in each space. The biggest challenge right now is how to sequence them, and never in my wildest dreams did I ever think that would be an issue in lung cancer, how to sequence multiple active therapies for patients. I mean, when I started, there was two drugs. There was carboplatin and there was paclitaxel. That was it. And that was 16 years ago. And man, we've come a long way. We've come a long way.
Yeah. Yeah, I mean, more options is always good, right? And it's incredible to see the pace at which discovery and advances are happening. So that's a win for our patient community. There's a question from the audience about how long, you might be dealing with getting treatment with ADCs. How long must you, what's the duration of treatment, if you will? Do we know that yet?
It's a good question. I mean, I would say this, that we use or leverage ADCs the same way we use or leverage targeted therapies or immunotherapy. is that we continue until two things happen. Either one, their cancer grows, or two, they're not tolerated. And I think the onus is on the physicians to really ask patients, are you tolerating this or not? I mean, as you saw from my scheme at the end there, quality of life is really important for me. If we're giving patients drugs and we're extending their life or curing them at the cost of misery and what their perception is of quality of life, we're not winning. And so we usually give these drugs, and I see patients every 3 weeks. And I say, how are you doing? And it's interesting because it's usually the patient's caregiver who's more honest than the actual patient, which is very helpful. But we'll give them until we can't give them anymore.
And so, kind of follow up to that question is, these drugs are relatively new. So, what do we know yet about resistance mechanisms? And we talked some about, especially in the setting of target therapy, transformation to small cell being a resistance mechanism that can happen. Since this is kind of a combined, approach with chemotherapy and target therapy, do we expect to see that small cell transformation, or what are the resistance mechanisms? Do we know anything yet?
Not yet. I mean, these drugs are so new and it's become so complex and nuanced, but this is where we really need to go, especially on the heels of what you just said, which is that we've got trastuzumab deruxtecan, and all of a sudden we've had these two new oral agents that we heard about at World Lung that look incredible. And the question is, are these drugs for a particular type of resistance mutation after Enhertu, or is Enhertu specifically active for a resistance mutation that occurs after giving zongertinib? So, I mean, this is the whole story with Tagrisso and how it came about, right? We had patients that were EGFR positive that received patient's tumor was EGFR positive, received erlotinib, and then had cancer growth. And then we found out that this T790 gene was the most common type of resistance, and that's where Tagrisso worked. And so this is how, understanding these resistance mechanisms is really critical to even understanding frontline decisions. So we've got a long way to go.
So going back to trastuzumab deruxtecan, there's a question just about how we determine if a patient is a candidate for that. And my understanding, I hope you can clarify, is obviously NGS has a role, but then we talked about the different levels of HER2 expression, or the different kind of classes of HER2 alteration. And so NGS plus IHC, what are we talking about to select these patients?
It's so complicated. Because I don't want to get too in the weeds here, but HER2 alterations or HER2 things in lung cancer can be measured in 3 different ways. It can be measured by what NGS, next generation sequencing, and that will measure it. It can also be measured by just something called IHC, which is the overexpression, or it can be measured by something called FISH, which is amplification. So there's 3 different HER2 pathways going on in lung cancer. It's the mutation, it's overexpression, and it's amplification. And incredibly, those 3 have nothing to do with one another. I wish they did, but they're totally different, even though they're HER2. What we know about lung cancer right now for Enhertu is that Enhertu works really well if you have a HER2 exon 20 insertion mutation. Again, so complex, but there's many, many types of HER2 mutations, and the ones that it really works for are the HER2 exon 20 insertions. That's number 1. More recently, 3 months ago, we found out that it also works in patients who have HER2 overexpression by protein level. So what we do now at Hopkins is we do both tests. We do the next generation sequencing test to try to identify the mutation, and then we also do the protein test to see if they have overexpression. I have yet to see an overexpressed lung cancer patient, but we're still looking. But there are going to be new platforms that are going to come out that are going to be able to measure this better. It's so confusing. And so this is, again, the part of even physicians call me and say, I've got this, and this. Can I use Enhertu? And I go, no. And they're like, really? So, I mean, you just have to be very careful when you're looking at the testing from the tumor.
And so I realize that I'm guilty of kind of lapsing into acronyms that may not be familiar for our listeners. With IHC, immunohistochemistry, the idea of staining for those proteins on the cells. So that's a way of kind of detecting how much of that protein is present. So another question from the audience: are ADCs chemotherapies is the chemotherapy delivered with more precision than existing chemotherapy? So I think the answer is yes. But
Yeah, I think that's the hope, right? It's just making sure the chemotherapy gets to the tumor really exquisitely and personalized to the level of the tumor.
Right. If the drug is delivered to the cancer cell, again, that's the idea that you want it to be specific to those proteins on the cancer cell. Why are other cells affected?
Yeah, great question. We're still trying to understand this, and what we've learned is that number one, it's not a perfect system. That antigens that we think are just on the tumor cell happen to also be expressed on other normal tissue. So something like HER2 mutations, something like TROP2 is an antigen that we're looking at, and these drugs that I showed you will focus in on TROP2 and kill those cells that are TROP2 expressed. Well, we have now found out recently that the gut or the oral mucosa of your mouth also can express low levels of TROP2, so you may have side effects there. So that's one thing. The second thing is, even if you don't have a target, sometimes the payload gets prematurely released in the circulation, and before the antibody gets to the target. And when you have premature release, that payload can go anywhere. And so that's another thing that we're working on, is making sure that the payload stays stuck to the antibody until the antibody gets to the cancer cell.
Mm-hmm. Yeah, so a lot of complicated, complex biochemistry involved in the linkers and the selection and how you build these, right? You alluded to some of the challenges around that with the one manufacturing issue. So, Dr. Liu talked a little bit about ADCs in his presentation on small cell, but a question came through of, are ADCs in development for lung cancers that haven't seen as much progress? So squamous, and we heard a little bit about small cell, but in case viewers weren't listening then, maybe you can talk about the ADCs for small cell.
Yeah, I mean, there's a few that are coming out. We had a small cell section of ADCs at World Lung, and all of these compounds are early on, but looking good. Small cell has been an unforgiving disease, and Stephen's kind of front and center with this and has led the charge globally on looking at these new compounds. And so I think there's going to be a lot of traction here. What's also interesting, and I don't want to confuse people, but ADCs can carry a payload, and they're also monoclonal antibodies that don't carry a payload, and those are called bispecifics. So with the payload, antibody-drug conjugate. Without the payload, they're called bispecifics, and believe it or not, these bispecifics look just as good. Now, you may ask, why would a bispecific work without a payload? You just told me, Ben, that it's all about the chemotherapy being delivered to the level of the tumor. Now you're saying that there is no payload, but these drugs still work. And this bispecific stuff is happening in small cell and squamous cell a lot right now. And so I know the nomenclature is so wacky, but if you could just remember ADCs, payload, bispecifics without the payload, but for some reason they both really work. What a time to be in the field.
It may be helpful. I mean, one way I can think about it is ADC has C in it, chemotherapy. So the ADCs have the chemotherapy payload, and then the bispecifics are really a way of tethering the cancer cell to those immune cells. And we know one of the great skills that the immune cells have is they can burst the cancer cells, so they can destroy. They have a lot of power in of themselves aside from that payload. So really exciting science and technology. Another question, you alluded to this a little bit, but are there ADCs in development for those with EGFR exon 19 mutations with no PD-L1? I think there's lots of ADCs.
Those are the most exciting advances. Yeah, sorry about that. Yeah, there's no doubt, and I didn't have time to go through this, but there are a few ADC compounds that we are testing and that are also coming out that look too good to be true in the EGFR space. And we'll have to see how this data pans out. These drugs don't have names yet; they just have letters and numbers. Several companies are looking at these, and I'm really excited about that. I'm really excited about the ADCs in the EGFR space. Patritumab deruxtecan, that one I mentioned, is going to be approved in the EGFR space one day, just when the manufacturing issues get sorted out. But there will be others that will be just as good, if not better.
Another kind of interesting, tricky question. Are ADCs unable to kill cancer stem cells? So,
It's tough to know. I
first,
mean, the
maybe you
question
want
is,
to explain what a cancer stem cell is for the audience.
yeah, I mean, cancer stem cells can be looked at in different ways, but a cancer stem cell is essentially the progenitor or the original cell that sort of makes things evolve over time. And a cancer stem cell may not have the antigen that the rest of the tumor has. So when we look at lung cancer, we really don't think about cancer stem cells all that much. We're looking at the cancer as a whole and how to beat it. And unfortunately, cancers as a whole and lung cancer have a lot of heterogeneity and a lot of different cells that have a lot of different proteins. So we want broad applicability of the ADC, but we also want to select a target out that's on all the cells.
Mm-hmm. Yeah. Well, listen, this has been an incredible session. I think we got through most of our audience questions. It's always great. You explain things in such an accessible way, and we really appreciate your time on a Friday afternoon on the heels of a major conference. There's a lot going on in the lung cancer community the last couple of weeks. So we appreciate your commitment to the lung cancer community and to our listeners. And thank you for your time, Dr. Levy.
Thanks for having me. It's great to be here.